Ladies and gentlemen, I'm Yuan from the Nuclear Medicine Department of Peking University Cancer Hospital. It's a great honor to share and discuss my current research with you. Today, I'm sharing a research article titled Contraction and Preclinical Evaluation of a zirconium 89 labeled monoclonal antibody targeting PD-L2 in lung cancer that we published in Biomedicine and Pharmacotherapy. Next, I will present the background, results, conclusion, and discussion of this research. First, let's briefly introduce the mechanism of immune checkpoint inhibitor therapy. PD-1 is a membrane receptor protein mainly existing on the surface of immune cells. PD-L1 and PD-L2 are the ligand proteins of PD-1. After the receptor binds to the ligand, the signaling pathway can inhibit T cell activity and proliferation, weaken the role of T cells in the tumor microenvironment, and ultimately enable the tumor to escape from the immune system. As a treatment, especially PD-1 targeted therapy aims to activate T cells and upregulate immune function by blocking PD-1 signaling pathway, thus ultimately achieving tumor clearance. However, how to screen potential beneficiaries of ICR therapy and evaluate its efficacy has become a current clinical challenge. Multiple prospective studies have confirmed that patients with high PD-L1 expression are more likely to benefit from ICI therapy. So, the FDA has used the immunohistochemical qualification of PD-L1 expression as one of the screening criteria. However, in recent years, with the deepening of research, multiple studies have confirmed that PD-L1 negative patients can also benefit from ICI therapy, and the therapeutic effect is not different from PD-1 positive patients. This has led us to deep reflection, and the search for new biomarkers seems to be urgent. Through our in-depth research, we have focused our attention on PD-L2, another ligand of PD-1. In fact, PD-L2 has been widely studied in recent years. Its mechanism is similar to PD-L1. PD-L2 on the surface of tumor cell suppresses T cell function by binding to PD-1 on T cells, enabling immune escape. Notably, PD-L2 and PD-L1 cannot bind with PD-1 at the same time. There is a competitive binding relationship, and PD-L2 is considered to have a higher affinity for PD-1 than PD-L1. Through our exploration of online databases, we have found that PD-L2 is expressed in more cancer types, especially in urinary malignancies, whereas positive rate was higher than PD-1 and was associated with tumor grading. In addition, several studies have confirmed that high expression of PD-L2 was associated with poor prognosis and survival of patients. A study published in the Clinical Cancer Research Journal suggested that the expression of PD-L2 might even be a better predictor of therapy response than PD-L1. Subsequently, some researchers think that the therapeutic effect of PD-L1 monoclonal antibody depends on the expression of PD-L2. Although there is currently no development of PD-L2 targeted drugs, some basic studies have confirmed that PD-L2 monoclonal antibodies could significantly inhibit tumor proliferation and progression, which provided new targets for the development of novel drugs in the future. In summary, we have developed novel PET tracers targeting PD-L2 for in vivo, dynamic, and non-invasive qualification of PD-L2 expression, so nuclear medicine molecular imaging, which will ultimately enable precise screening of potential beneficiaries of ICI therapy and evaluation of therapeutic efficacy. In previous studies, we have constructed a tracer labeled with radioiodine 124. In this study, we modified the tracer labeled with a radioactive mental nuclide, the zirconium-89, to achieve better imaging effect. Next, I will present the results we have achieved. Firstly, we constructed trafficked cells with high PD-L2 expression and screened for wild-type cell lines with high PD-L2 expression through Western blot. Subsequently, we established a tumor-bearing mice based on the above positive cells for in vivo evaluation and imaging. Immunohistochemistry and HE stain confirmed that the successful establishment of the PD-L2 high-expression tumor-bearing mice model. The process for conjugating ATL2 to DFO and DFO-ATL2 labeled with the colon 89 was shown in figure 1A. The radio tracers, the colon 89 DFO-ATL2 were successfully prepared. They maintained high yield and radiochemical purity and met the criteria for further evaluation. Then, we performed relevant biological evaluations. Radio-ELISA results showed the tracer had a high affinity for human PD-L2 standard protein. The clearance of antibody tracers was also relatively slow. For cell uptake assays, the uptake of the tracer in the positive line A549 PD-L2 gradually increased with time and was higher than that of A549 cells at each time point. After that, we conducted in vivo imaging studies. First, micro-PET CT imaging of normal cat mice was performed. The imaging and LI results show that the heart and liver uptake values were higher at 3 hours. With increasing time, the signal of the blood, pore, and liver gradually decreased. Other tissues such as the brain, muscle, and bone maintained low signals. As shown in figure, the tracer achieved good imaging effect in the positive mice model. The uptake of the tumor area gradually increased with the extension of imaging time, reaching a peak at 96 hours, which were significantly higher than other groups. For the analysis of the ROI shows that the tracers in A549 PD-L2 group showed a higher tumor-to-non-tumor ratio. In addition to our trafficked model, the wild-type model can also achieve better imaging. For these tumor-bearing mice, the tracer was specifically taken up in the tumor area and showed desirable rotation over time. By our analysis, imaging achieved a higher tumor-to-muscle ratio at 72 hours and a tumor-to-brain ratio at 168 hours. This is the conclusion of the research. The tracer was generated at high radiochemical yield with high purity. It showed a high affinity for PD-L2 and selective accumulation in PD-L2 positive tumors, resulting in a high tumor to background ratio for delayed PET imaging. So all results demonstrated that the corner 89 DFO-ATL2 has great potential for detecting PD-L2 and evaluate therapeutic effects. Let's have a brief discussion next. First, PD-L2 can bind not only to PD-1 but also to RGMB, and the overall role of PD-L2 in the tumor microenvironment need to be revealed in future research. In previous studies, we constructed a molecular tracer targeting PD-L2 for the first time and carried out preclinical evaluation, confirming the feasibility of this PD-L2 targeting tracer. Compared with previous studied tracers, radioactive meteonuclear zirconium-89 improved imaging of tumors, and it could also simplify the preparation steps. Finally, iodine 124 labeled tracers may not be appreciated for detecting stomach cancers and urinary malignancies because iodide is cleared was in urinary system. There are several limitations to this study. One, for clinical translation or for humanized antibody is needed, so ATL2 needs to be further humanized. Two, for antibody tracers, slow clearance in vivo is an important factor limiting clinical translation. 3. We found a decrease in the affinity. We think the reason may be that the non-site direct binding of DFO affects the binding site or steric conformation of the antibody. That's all. Thank you for listening.